All right, so let's go ahead and write a to-do list very quickly. What we've said already. Number one, and I shouldn't do it in purple, I suppose. Number one, you have a video for DNA project that you're going to watch and take notes on. You better hurry up and write the notes, or you're going to have to catch up on the video because I got to get going. Thank you. So you have to watch a video, and the link is there in the handout. I just handed it out, and it's also posted on Schoology. So there really is no excuse for not getting the video done. It's going to be very informative. The questions are deep, and I will be grading them for content. So, two, you can pick up instructions and the model cutouts tomorrow morning, or after, bless you, fourth period, or after fourth period today. So if you want to start early, you can. This, I want to be absolutely clear, this is due Wednesday, and the model itself will be due on lab day. For you guys, lab day is Thursday. For another class, it'll be Friday. So are we clear on that? There's no questions. And for these two and from all assignments from now on, as we said, it's going to be -10 for 1 day late. -20 for 2 days late. And 0 from then on. So I'm going through this with you because I don't want any questions or concerns or I didn't knows and I wasn't sure's and etc. Okay. Also on your syllabus, I made little circles with due dates. You should know that your lab report from last week is due Wednesday. So make sure you're getting that lab report done for Wednesday as well. And of course your chapter reading is due today, end of day. Chapter 1 and 6 was due today.
It's not 136?
Everybody says that. How is it confusing? When I say 1 and 6, I've been saying the same thing literally for a month and a half.
The emphasis on the and keep on disappearing in my head.
I know. Thank you. Thank you so much for being so honest. The emphasis, she said the emphasis on the and keeps disappearing in her head. Okay, that's fair. I can accept that. My thing is, you got to get, it's 1 and 6. And if you did 1 through 6, good. You didn't lose out. You did work. You read stuff that we're going to cover. You're ahead. When we start doing those other chapters, you already read them.
So we didn't have to do all
I'm not talking about that again. I can't say it again. I mean, I'm speaking English, I think. Sometimes I might slip into Spanish, but it's 1 and 6. And, not through.
So I have a question. If the notes 1 and 6 were due today, why are you still looking at number 6?
What did she say?
Like on the syllabus?
Why are we still on number 6 on the syllabus?
No, I mean, are we supposed to be taking notes on anything? We've done chapters 1 to 6
Are you supposed to what?
Are we supposed to be taking notes on any chapters since we already finished the 1 and 6 notes?
Okay, great question. So, you were supposed to read 1 and 6 for this entire unit. The entire unit. You only had 2 chapters to read. That's it. That's all you had to do. A month and a half. Read those 2 chapters whenever you wanted. Get the notes done and turn them in. I have many people that still haven't done them. And we're still working on chapter 6. There's a lot to it. I'm adding material that's not in the book. If you're not taking notes, you're going to be lost. So you have to take notes in class. Chapters 1 and 6 in the PDF versus the book, the Zebra book, they're about the same. There's some differences because they're different versions, but they're about the same. All right, go ahead with your questions. This last question. Go ahead.
So what on the thing say do Monday, September, like do today, but then the description says turn about Wednesday?
Where does it say turn in by Wednesday? It says chapters 1 and 6 due today.
Let's consider exclusion.
What does it say in Schoology? It says turn in by Wednesday? Is that what it says in Schoology?
What's today is Monday.
Huh?
Today, but
Wednesday what? What date on Wednesday?
But you said please turn about Wednesday.
Yeah, here's why it says that, and I'll make it. I'll change that after third period today. This is the last thing I'm gonna say. It's the reason it says that is because they were due last Wednesday, and actually they weren't due last Wednesday. They were due the Monday of that previous week, and actually they weren't due that week. They were due the Monday of the previous week, because 4 times I moved it back. The first time I said, okay, turn them in next week, Monday. Then the next time I said, okay, turn them in Wednesday. And then Wednesday, you guys, a bunch of people didn't turn them in, and we had grades due, so interim progress reports due. So I said, okay, we'll turn them in on Monday, which is today. So I changed the date
So it's
4
not,
times.
it's not satisfactory, unsatisfactory performance no more, it's the actual grade.
Yeah, now we're going into the full-on grade. And you have a test coming up. A, B, C, D, or F is what you got. Tests are coming up about this material. I'm glad we're having this conversation, but we really got to get moving here, or you're not going to be ready for your test, and all kinds of problems are going to come from it. Yeah? Say what?
When is the test?
Next Friday, next lab day. I wasn't going to decide. I really wasn't going to make a choice, but you made me make a choice, so I made the choice. Next lab day. All right. Okay, so that's where we're at. So chapters 1 and 6 are due today, and I will change the description in the assignment. Now, can we get on with content? Let's get on with content. So today we're covering proteins. Let's talk about this idea of polymers and monomers that we had started last week. Each of the biomolecules have different monomers, have different building blocks. So for the polymer, let's be clear, lipids are not, and I'm going to write this out on the out here, lipids are not polymers. And we'll talk about what they are later, but they are not polymers. So that leaves you with 3 biomolecules, and the 3 biomolecules that are left are proteins, which we'll talk about today. We're going to talk about, which we're finishing up today because we did our enzyme lab last Friday. You should have written it up. If you have any questions about proteins, today might be a good day to ask it in case you need to finish up lab. You should have all your answers for your lab write-up done already, but if you have any final questions, today might be a good day to ask. The monomers for proteins are amino acids. Then you have carbohydrates. The monomer for carbohydrates are monosaccharides. Saccharide means sugar, mono means one. So monosaccharides are monomers for carbohydrates. And the monomer for nucleic acids are nucleotides. We're going to go into the details of this last one on Wednesday, right before you do your lab. But before we do, I'm going to finish up proteins today. I'm going to introduce nucleic acids so you have some idea what you're doing in case you want to start building the polymer of nucleic acids today or tomorrow. Then tomorrow we'll talk about carbohydrates and lipids. All right, these two we're going to talk about together at the same day. And then Wednesday we'll talk about nucleic acids. So if you remember for proteins, when we talked about proteins, we said there's monomers, and the monomers of proteins are these carbon with a hydrogen, has this R group, we said is a variable, and we said it has this amine group, as an NH2. You should be, at some point, you should be able to draw it as easily as I'm drawing it here. Carbon, double bond O, single bond OH. That is one amino acid. When you're going to put them together, they're going to face each other in this direction. And we'll talk more about amino acids. It's a very important topic. How do we put these together? Does anybody remember? Anybody have in their notes? We put together all biopolymers the same way. It's some kind of synthesis. It's dehydration synthesis. What happens is this carbon, if you're going to put it together with this block, and you're going to put it together with this block, this mono or amino acid, you're going to put it together with this amino acid, you have to link them. This carbon can only make 4 bonds. It's done. It's full. It can't make any more bonds. 1, 2, 3, 4, right? This can only make 3 bonds as 1, 2, 3. Everybody's done. It can't make any more bonds. They're neutral and they have a full octet. So what happens is that you have to get rid of something. You have to exchange something. That's called a chemical reaction. An enzyme does this. There's an enzyme that does this. The enzyme is actually made of RNA, and it's called, why is this thing still up? And it's called a ribosome. A ribosome is made of RNA. It's one of the few RNA enzymes. That's going to put this together. You're going to take 2 amino acids and bring them together. Yeah? I want you to figure it out. I told you in class, but it's in the book, but I'm going to go ahead and say it, and hopefully I'm going to give you all the components, enough of the components that you should be able to say it out loud. So you got to make room for it, and so what's going to happen is that you're going to take these two are going to leave, and that's going to leave some open bonds that will allow them to connect. What do you think those two are going to form? Water. That's right. So water is going to come out. It's going to lose H2O, lose water. So the process is called some type of synthesis. What's it called? What word are you going to use? That's right. Somebody remembers. Dehydration. Synthesis. Some of the books might call them condensation reactions. All right. Both words make sense. right? Your water being taken out, dehydration. Condensation is when you form water. So that makes sense. When you build muscle, you make water in your body. When you build proteins, you make water all the time in your body. You take OH and H and you produce H2O. So when they're put together, they look something like NH2HC. This is R1. It could be one of how many different amino acids? How many different amino acids can you have? Say again, I can't hear you. 4. Somebody guessed 4. That's not right. I told you in class, how many different R groups can you have? How many different? Not 2. Not 3. 20 different amino acids. 20 different amino. Guys, if you're not studying, what if I had a quiz today on this? You had a lab on Friday. If I gave you a quiz today, sounds like people wouldn't do that well. You guys got to keep up with this material. That's why you should be doing the 6 R's every day. A little bit every day gets you a lot further than trying to cram it the last minute. A lot less stressful too. So that could be R1 or R2. And you can keep adding them, you see. Just keep adding them here this way. And make this long chain of amino acids. And it can be long. Now these chains, there's these 20 different amino acids. Some of them are hydrophobic. Some of them are hydrophilic. Some of them are acidic. And some of them are basic. Some of them have rings. Some are big. Some are big and some are small. So there's all different kinds of amino acids, 20 different ones. And those 20 different ones give the proteins that they make, the polypeptides that they make, and then the proteins that, therefore, that form afterwards, different properties. Yeah.
Can you make that big reference?
Which part?
That part right there.
I mean, when you see that part, you mean this here or this here?
That.
This?
No.
Yes. So hydrophobic, hydrophilic, and I'll do this. I'll highlight the hydrophobic with yellow, and I'll highlight the hydrophilic with blue. Hydrophilic means it loves water. Hydrophobic means it hates water, in case you've forgotten. Acidic I'll make red, and let's see. Basic I'll make green. All right. Now they could be more than one. They could be hydrophobic and have rings. They could be big and be hydrophobic, or small and be hydrophobic. So there could be all kinds of different combinations here. So we call a bunch of these put together a polypeptide. And the reason we call it a polypeptide is because this combination, this bond, this bond, oh, I hate that. This bond between the carbon that we just formed between the carbon and nitrogen, you see that? This bond we just made here, this is called a peptide bond. It's a peptide bond. So that peptide bond, a bunch of them put together, a bunch of these amino acids put together, we have a lot of peptide bonds, we call it a polypeptide. Does that make sense? Yes or no?
Yeah.
Who said no? All right, what part of that does not make sense to you?
So the bond that we did is a polypeptide.
Say that again.
The bond that we did, like
The bond that you just made is called a what? Look again.
Peptide.
Called a peptide bond. I think that's pretty big, but if you need it a little bigger, does that help? Is that big enough for you? Peptide, yes? You see it? All right, so that's a peptide bond. A bunch of them put together is called a polypeptide. More than one of those. More than one bond. So you can have this long chain of amino acids. You can have you can have a basic one connected to hydrophobic one, another hydrophobic. The basic one could be, this hydrophilic. There'd be two hydrophilics in a row. There could be, the basic one could be also be hydrophobic. There could be another basic one that could be hydrophobic. Then you could, do you see what I'm doing here? Do you understand that there's each one of these? This is one amino acid. This is two amino acid. Let me do different colors here. This is one amino acid. This is two amino acids. This is three amino acids. Each one is connected by a, what's each of these connected by? A peptide bond. A peptide bond. Not a peptide. A peptide bond. And then you have amino acid 4, and this could keep going. Some of you are listening, some of you are not. I hope you're listening I hope more of you are listening than are not. So if these are all hydrophilic, they like water, 4, 5, 6, and 7, all connected by peptide bonds, peptide bond, peptide bond, peptide bond, and peptide bond. And then you have a bunch of R groups that are, remember, now a bunch of R groups that are hydrophobic. And then there's a bunch of R groups that are hydrophilic again. Again, each one of these is connected by a peptide bond, peptide bond, peptide bond, peptide bond, 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 bond. So that's just a chain, like I don't know what, like knots in a rope, all in a nice. They make the rope, and each one of these is a different amino acid. So there's 7, there's 8, 9, 10, 11, 12, 13, 14, 15. Then there's 16, 17, 18, 19, 20. There's 20 amino acids in this polypeptide. This is called a polypeptide, and there's 20 amino acids. They're not all different. You have this group here is hydrophobic. Do you see the color chart? So this group here is hydrophobic. This group here is hydrophilic. This is hydrophilic. Then you got a little bit of mixture of both, one hydrophobic and a lot of hydrophilic. So this is in what? What is this made in? What's the solvent of life? What is all this always happening in? Water. Excellent. It's always happening in water. If this thing is in water, what are these hydrophobic, water-hating amino acid side chains? I want to reiterate. I'm going to say this again. All the amino acids are the same. The difference is this R group is different in each of them. It could be the same if they're the same amino acid, but there's 20 different amino acids. So what are these hydrophobic amino acids going to do? What are these hydrophilic amino acids going to do in water? What's the water-hating ones? These yellow ones, the hydrophobic. What are they going to do?
They're going go against the water.
They're going to go away from the water, and the hydrophobic ones are going to do what? They're going to go towards the water, right? They like water. So this is going to do something called folding. It's going to fold. A lot like what you're going to do with the DNA molecule, it's going to fold. This protein is going to fold. And I could ask you to go ahead and fold it, I suppose, but I'm just going to, for argument's sake, I'm just going to go ahead and fold it. And what you're going to get is a bunch, the yellow ones forming the inside, and the blue ones are going to form the outside.
So they'll fold it in the middle?
Say that again?
They'll fold it in the water?
They'll fold in the water. Yeah, that's right. They'll fold in the water. What's that? Is that lightning?
No, I think you took a picture.
Oh, somebody took a picture. So this polypeptide's going to fold, and I could number them. You know, you could sit there and say this is what? Maybe that's 1, and then 8, 9, 10, 11, 12, 13, 14, 15, or whatever. And then you have on the outside, you have maybe this side 16, I'm sorry, it would be 16, 17, 18, 19, and 20. This is obviously horribly, it's not to scale, but it'll have to do. And then maybe you have one little yellow one that sits inside, that's number 1. And then you have 2 and 3, and maybe this one sits on the inside, and you get 4, 5, and 6. So you see it folds up. The chain doesn't break. It just folds up and makes a shape. And so what happens is you have this internal part and then this external part. So now it has a function. It could do something. Maybe it's an enzyme. That's one thing proteins do. Maybe it's an enzyme like the one you used on Friday, catalase. Maybe this hydrophobic part, maybe this is the active site. Maybe that's the active site of the polypeptide. Maybe it's one component, one chain of a group of polypeptides that come together to do something. There's a polypeptide that's very famous, maybe you've heard of it, it's called hemoglobin. And it has one long chain of alpha chain, it has actually 2 alpha chains. So 2 chains that are exactly the same. And then it has 2, this would be the alpha. And then it has 2 beta chains. Exactly the same.
Isn't that hemoglobin?
That's what I just said, hemoglobin. So hemoglobin is actually 4 polypeptides. So a protein can be made of more than 1 polypeptide. Or it could be just 1 polypeptide. All depends. Hemoglobin has this little heme group. Looks like a little square. I should do a different color just for the argument's sake. Has this little group here, at the center of which is this big iron atom, Fe. This hemoglobin carries oxygen. Carries oxygen gas from your lungs through your blood. It makes your blood red. Makes your blood red. Mutations in these polypeptides lead to sickle cell anemia. So change one amino acid in one of these strands, alpha or the beta, and now this hemoglobin doesn't work very well. Change it one way, you get thalassemia. Change it another way, you get sickle cell anemia. So that's all. That's all it is one chain, one polypeptide change, one R group changes, and that's it. Yeah. Yeah. Guys, you can go to the restroom anytime. Just grab it as long as the pass is there. So hemoglobin is actually important for carrying oxygen. It's not an enzyme, but yesterday we talked about a protein called catalase. And not yesterday, but Friday or Thursday when we did our lab. Catalase is an enzyme that gets rid of hydrogen peroxide, takes H2O2, and turns it into oxygen gas plus water. Very important reaction found in almost every living creature on the planet. And we have other proteins. There's these long proteins that lay kind of in strands, and they overlap a little bit. What do you think? What do you think this is forming? Say what? As they lay down on top, these proteins lay down on top of each other, what do they form? Skin. Yeah, this would be skin. And there's another protein that kind of does this. This is a protein, not DNA or anything. Kind of, they wrap around each other. Yeah, that's right. This would be hair. And if there's a lot of sulfur, then the hair does this because the sulfur attracts itself. And so, that's what?
It'll make it curly.
It makes it curl up. The more sulfur you have, the more disulfide bonds. So you have hair of different kinds. Then some proteins form these sheets. What do you think this looks like, these proteins that form sheets? Hard, see-through sheets. Nails. This is made of protein right here, your nails. So skin, hair, pigments. We talked about pigments like melanin. We did that when we talked about chromatography. We said there's pigments that absorb, they're made of proteins. We separated the pigments during when we did our chromatography lab. There's also proteins. So we got structural proteins. So they can be enzymes, they can do function, they can actually do a function, they can be structural. They can be used for transport. They can be enzymes. Say that again. I'm sorry, the fan is.
I said, what was this Jew for structural enzymes? Or no, not structural, functional.
Yeah, these are structural proteins, right? So these are all structure. They make us look in a way. They hold us up. Your bones are made of a lot of different proteins. The structure of each of your organs is made of proteins. Obviously, they can be used for movement. All your muscle is made of protein. Well, a lot of protein. There's other stuff in it. So there's a lot. Proteins do a lot in your body. One of the more. In fact, we thought for sure. When I say we, humanity, scientists, thought for sure that proteins were what held the information of who you were. The stuff you inherit from your parents, we thought it was proteins. I mean, which would you rather, let me talk to you a little bit about DNA and RNA, because some of you might want to try that lab. So that's it for proteins for now. We'll come back to them. But do you understand that? What is it you have to know for your test? You got to know this structure. You got to know what an amino acid looks like. You have to know that this is a peptide bond. You have to know dehydration synthesis. You have to draw it. Now you will. Now you'll have to draw it. And then you'll have to draw the amino acid, the general structure. That will be one of the things you're going to have to do on the test. You'll have to... You have to identify peptide bond. You have to know what a polypeptide is. It's a bunch of amino acids put together by peptide bonds. You'll have to understand that they fold. You don't have to describe why they fold or how, but you have to understand and say they fold. They form specific structures. They can form enzymes. You have an example. If I ask you for an example enzyme, you have at least one. You should know very well. They transport things like oxygen. In the case of hemoglobin, they involve themselves in structural things like nails, skin, hair, and pigment. They can absorb light in pigments. They absorb. That's what they do as pigments. Absorb light. Yeah. I'll ask for all of them. I'll ask for examples of each of these now. So you have a movement. We will have a review day where you can ask a lot of these questions, guys. So you do want to know all your examples, because I could, even if I don't ask for all three, how do you know which one I'm going to ask for? You don't. You got to know all of them. All right. So structural transport, enzymes, movement, muscle for movement, and they absorb light, and pigments absorb light as well. So all proteins do all that. Now nucleic acids, they're made of a special sugar called ribose. There's a sugar called ribose. It's a 5-carbon sugar. It forms a ring. Pay extra attention because I'm about to give you some numbers that you need to know. You don't need to know them right now, but you will need to know them. Does everybody see that this oxygen is an oxygen atom? It looks like 0 on a clock. Yes? If this were a clock, this is 0. Where would you put 1? On the right. So this is our 1, this is our 2, this is our 3, this is our 4, and this is our 5. In all of organic chemistry, when you have a ring, that's how you number them. Start with the oxygen, go clockwise, and number them. 1, 2, 3, 4, 5. Now that's the ribose. Now it could be ribose or it could be deoxyribose. And I'm an idiot. I spelled I. It should be Y, I think. So deoxyribose, on the second position, this is going to be key. There's 1, 2, 3, and 5. Those are the 3 important positions you have to know. On the second position, it either has an OH, a hydroxyl group, or it just has an H. One or the other. If it's missing the H, if it's missing the oxygen, which of the two is the sugar? If on the second carbon it does not have if it has just the H, no OH, it's deoxy. It's deoxyribose. So it's that second carbon is going to be the key to understanding if this is deoxy or if it's just plain ribose. The difference between DNA and RNA. It's one of the first differences. So the third carbon is going to have a hydroxyl group, an OH. And the fifth carbon is going to be connected to one of the other things that makes a nucleotide, that monomer of DNA and RNA, is phosphorus with 4 oxygens connected to it. This big old thing is called phosphate. That's one of those functional groups. Is it time to go? Is that why people are moving around? What time is it? Is class over?
8:55.
All right, so I got time to take attendance. All right, so the last piece before I let you go. So there's 3 pieces as you're looking at this DNA video. Phosphate, you're going to deoxyribose without an oxygen, second carbon. And at this first position, before you put stuff away, you have a nitrogenous base. Those bases are as follows: A, T, G, or C in DNA. A, T, G, or C. You're going to be building DNA. Let's focus on A, T, G, or C. Each of these is slightly different. Pay attention when you read about it or see it in the video. Let me quickly take attendance and you're out of here. I guess I watch more elevated versions. But anyways, when you have nitrogenous bases, you're what we call a nucleotide. You're going to be building a bunch of these, putting them together. A nucleotide is your deoxyribose in DNA is deoxyribose. This is DNA. That means no oxygen here, just an H. Here, there's other stuff to this, but we'll talk more about that I'm just going to put a phosphate, a P for phosphate. And here you have this nitrogenous base. It could be a single ring or it could be a double ring. but they have this nitrogenous base. It's either A, T, G, or C. So this is either A, this can either be A, T, G, or C. And that's it. 3 parts to this. 1, a phosphate. 2, a deoxyribose. 3, a nitrogenous base. You're going to take a bunch of these, and remember I told you there was another important part, this third carbon. This third carbon is going to connect to a phosphate again, which is going to connect to the 5th carbon, which is part of the ring, which has on it a nitrogenous base, which is connected to another phosphate. So there is your repeating units, your monomer. Here's a bunch of monomers, monomer 1 and monomer 2, connected one after the other. Monomer 1 and monomer 2 connected to form a polymer. That's DNA. And you will be building that over the next couple of days, and we'll talk more about it on Wednesday. Then we take attendance and we're done. So you can't force these molecules together. So just like a jigsaw puzzle, they fit in a certain way. And the only way these fit is A always fits with T. And G always pairs with C. Now there's all kinds of great experiments that went on to prove this, but you can just memorize it. A goes with T and G goes with C. Now some of this information should help you when you're trying to answer your questions on your videos, and certainly when you're trying to answer the questions on the model building. Please understand there are questions you have to answer, measurements you have to take when you're building that model. It's not just building a model, it's building it and thinking about how the DNA structure gets put together. So A gets put with T, and this keeps going. This keeps going for a very long time. Say that again. So when you think about a DNA molecule, you're going to see some pretty short DNA molecules. Like we made this one in AP. It's relatively long, I guess, compared to what some of you might see. But these molecules, obviously they're tiny, right? You can't see them, they're molecules. They're something like what? 3 or maybe 5 or 10 or 20 atoms in width, right? So they're only about 20 atoms wide, but they can be 3 meters long. They can not well. Do you understand what I'm saying? That these molecules are very long. They have billions of billions of these nucleotides. Obviously we're not doing that in class because that would take the rest of our lives and probably another 100 generations. But what we're going to do is build a short segment of it. But you have to always keep in mind, you have to use your imagination. If you're not using your imagination, you're going to get these answers wrong on the test. Certainly if you're looking at each other's in the eyes instead of looking at me. So billions of nucleotides long means that this is a very, 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 very long molecule. And where do you find this molecule? You have to put this in perspective. Always try to keep scale in perspective. Where do you find this molecule? Where is DNA found? In the nucleus. So you have a cell that you can't see without a microscope. Is that correct? And then you have a nucleus, and then you have all kinds of organelles. And inside this nucleus, right in here, you have this DNA molecule. And you don't have just one of them. You personally, you have 23 pairs of these things, these DNA molecules. You have a total of 46 molecules, 3 meters long, all wrapped up and shoved in this tiny little spot in the center of your cell. Of every one of your cells has the same number, 46. We call these, and some of you may have learned it in middle school, we call these molecules chromosomes. So when you're talking about a DNA molecule, this DNA molecule is known as a chromosome. Now, a chromosome isn't a chromosome until it's all wrapped up. Obviously, if you're going to have all that DNA in that tiny little space, I hope it's obvious, you have to organize it, right? I mean, have you guys ever let your hair go with, I mean, if you have long hair, not your hair, because your hair wouldn't matter. But if you had long hair, have you ever had long hair or longish hair, what happens when you leave the hair alone and don't comb it? After it's nappy, it gets all tangled up. I try to comb my hair after, literally after just a day, I try to comb this long hair, and it hurts.
Your hair long?
Ish. Longer than I normally have it. It's not long to you?
I can't see it.
Oh, okay.
Turn
Well.
around.
You can check it out later. But so it gets all knotted, and when you try to comb it, it's hard to comb it, right? So the key is that you have to organize it. You have to continuously put energy in to keep it organized. And DNA, you have to do the same thing. So we have these things that we call histones, and you'll learn more about them later. But it's important you have some idea what they are. They're these proteins. They're kind of a block of 4, where you wrap DNA around them. They're like curlers for DNA. Does that make sense? And if you have enough of them and organized in the right way, if you have enough of them and you have them organized in the right way, you can have, the chromosomes will look something like this. And when you look them, you're going to look at them under the microscope, and you're going to see these things. They're going to look like this. They're going to look like they're going to look black, but I can't draw black on this black background. So they'll look real dark because that's the stain that we use. We use a stain that makes them look real dark. And there'll be 2 of them, obviously, I hope, because they're pairs, and we're going to see them right after they've been copied and they're being moved around. So they're going to be linked together. And we'll learn more about that when we go over mitosis and meiosis. And they're linked together by another protein. Some of you may have learned it in middle school. But that's what we call a chromosome. When we see the chromosomes, we see them this way. But when they're unwrapped, they're just long strands of DNA. Rarely are they completely unwrapped. Usually at least part of them are wrapped around some histones, because there really just are, there's just too much DNA in this cell to just let it all get tangled up. If they get tangled up, things are going to get mixed up, chromosomes are going to break. Cancer, right? That's what causes cancer. One of the causes of cancer. There are these chromosomal breaks, and aging. That's why we get older, because of this. This is the reason we get older. Our DNA breaks, and we can't fix it. Normally we fix our broken DNA, but as we get older, we have more and more breaks. They accumulate, and you start to mix up your DNA, and the cells don't work the way they normally would. So that's why the diseases of aging come upon us. So all that should be just for perspective. We'll come back to that. None of this stuff here that I just said will be on the test. That'll be on a future test. We'll go into details of that. But I just want you to make some connections now. So what's important here is that you understand that A goes with T, G with C, that there's 2 strands, that the 2 strands, what holds these 2 strands together? Let's be clear about something. These are all covalently bonded to one another. You see the covalent bonds here? These lines are all sharing electrons, very strong. But then there's hydrogen bonds between these. There's 2 hydrogen bonds between the AT, and there's 3 hydrogen bonds between the GC. And so GCs are actually held together stronger than ATs. But that's it. That's what holds them together, is these hydrogen bonds. So these are 2, it's like taking 2 Velcro straps. The straps themselves are pretty tough. You link them together, the Velcro holds. But you can rip the 2 strands apart, right? So it's the same thing here. These 2 strands can be separated. That's going to become important later when we're making copies of it or we're trying to read it. Because when they're together, we can't read it. You see that? You can't read this because the A and the T they're stuck together. And by the way, there's one more key to this, is that on this side, it's always opposite the other. So this, if that's 5', then this is 3'. So there's an OH group out here. And if that's 3', then this is 5'. And that's a phosphate over here. All right. So they're always opposite one another. So they're parallel, right? These are parallel. And they're opposite. So we call them anti-parallel. So that is a quality you need to know for the test. Are you literally fanning her?
She decided to do it herself.
All right, so you know what you guys can do is get the fan. There's a fan. That what you're doing is just going to make you hotter. Do you not know that? Do you not understand that? When you fan yourself, it just feels cooler, but you're spending so much energy moving your hand back and forth that you're literally you're actually making more heat than you're cooling off. The same with the fan, but the fan getting hot, nobody cares. So what we can do is we can put that fan in the window, in the doorway, point it, what I would do is move those papers, point it towards the people, because they're the ones that are dying of heat, I guess. You might want to move the paper before you turn it on. And then that should cool you off to some extent, much better than fanning yourself. But that means I can't hear you too well when you're asking questions, so speak up. Although no one's asking any questions yet, so whatever. So they're anti-parallel. They match up A to T, G to C. C has 3 hydrogen bonds. A and T have 2. Those are all very important. When they're wrapped up, they form a double helix where you get a minor groove and a major groove. Those are all kind of qualities of the structure of DNA. And the repeating units are made of phosphate, a deoxyribose sugar, and some type of nitrogenous base. The nitrogenous base can be A or it can be T. And honestly, if you paid attention, bless you, you should be able to answer these questions pretty clearly. So I'm going to run through these questions and see if does someone who had issues with this, does he have any specific questions? Yeah. Come up and get whatever you need, or ask somebody that already has them to come up and get them, so you don't, because you don't know what you're looking for.
You said it earlier, but what's the
Say that again. My dear children, I can't hear with the fan and... Okay, I'll wait. See, this is why I asked you to bring someone up here who knew what was going on so it wouldn't be an interruption, but that's okay. You know what you want.
Okay.
That's double yellow. You gotta get out the blue one. Oh, okay.
I wish you all would say, guys, and by the way, this is what the problem is with coming in late, number one. Number two, can we just get, I know you don't want to hear it. It's not your fault you're late, but this is why being late is an issue.
Can I have the double helix family? I misplaced mine. Thank you.
Can we get back to her question? What was your question, dear?
So you see the key box, right?
Boy, you start asking a question, somebody literally starts talking exactly when you start talking. Let's try that again. Speak up. The fan is in my ear. Go.
You see the key box, like, what does that stand for?
That, again, is, let's go back here for a second and take a look at it. That P stands for this. It's a phosphate. The word is phosphate. It's one of those functional groups you were told to memorize. Remember I told you there were there were like 6 of them. I told you what was important, what wasn't. I said these you have to know. Phosphate was one of those. The amine group was another. Hydroxide was another. Carboxyl was another. So I keep saying this, but if you memorize those functional groups, it will make your life a lot easier. Some of you are going to resist, I understand, but we'll move on. All right. So there's these two guys. There are these two guys named Shargaf and, well, I forgot the other guy's name, but there's this guy named Shargaf, and pronounced this, or spelled this way. I don't know if I'm pronouncing it correctly. If not, too bad. And they looked at DNA. Now remember, you can't see DNA. Please understand that in science we don't see things. We can't see them like you can see a jigsaw puzzle or a Lego block. All we can do is do chemical reactions and look at indicators just like you do in your labs, and we can run them on gels or like chromatography, right? We can see bands, but we can't see the actual molecule. Today we can, because we've used all kinds of imaging techniques, and thanks to a lot of really dedicated people, we have a lot of really great experiments that give us a good idea what they look like. But nobody can actually pick up a thymine and hand it to you like we can with these paper drawings. So Shargaf looked at a bunch of different organisms, and he said percent, I'm just going to go ahead and replicate the graph, the table here. Percent A, percent G, percent C, and percent T. I'm really not doing justice to this amazing man's work. He's one of the reasons we know what DNA is and how it, and the code, and we're able to read it. It's because of him. So, and all the men and women that worked in his lab. But when we look at this, and he said, look, there's 31.3% here in this animal, there's 32% A in this animal, there's 28% A here, 30 and 30. I'm just going to, I'm writing this quickly, so it's a little messy, I apologize, but we're just going to move on. Then this is 18.7%, and then this is 17.7%, and then 21.4%, and then 19 and 19. And then C, you find it's 17.1%, and 18.4%, and 21.5%, and 19 and 19. And then T's, we know it's 32.9%, and 32.1%, and 28%, and 29%, and 30%. I'm leaving off the 0.3s in this case. All right, so you're looking at this data. These are all different organisms. This is yeast, and our friendly yeast, right? And then this is sea urchin. That's and this is rat, and this is human, and this is another human. This is from the thymus, and this is from human sperm. So what we're dealing with here, what do you notice? Look at this data. This is data. Your job as a scientist is to find patterns in data. That's why we use math all the time. Math is the language of science. So when we look at this math, what do you notice? What's the pattern?
The numbers never go below. The lowest number is 17.
Lowest number 17. That's a good observation.
The highest number is 32.
So lowest and highest is always an important thing to look at. Lowest is 17, highest is
32.
32. All right? What else do you notice? Looking for a pattern. What do you notice? Watson and Crick, by the way, I really have trouble discussing stuff about them because one of the two ends up being a little racist, and they both stole their information from Rosalind Franklin. They stole her X-ray. I really have a hard time with that they stole this information from a colleague. And she was a woman in science at the time. Women were not welcomed. And these men treated her like crap, and then they disrespected her and stole her work. So I have a real problem with that. But they did figure some stuff out, so I got to give credit where credit's due. They used Chargaff's rules to help them discover this stuff that I'm teaching you right now. The stuff I'm teaching right now came from Watson and Crick. They're famous physicists, but again, they're thieves. And they're misogynists, and at least one of them's a racist. So I'm going to tell the truth. That's what it is, all right, as far as I know. I don't know them personally, but I heard them tell the story of how they stole this information from this woman and how they treated her. They felt bad about it later, but only after they got caught. So, all right, so somebody tell me what you see here. It's a table. What do you see? What do you see? What's the pattern? Give me a pattern of the data. If I'm asking you, is because you have a good chance of finding it if you're looking. You don't know it. You've never seen it before unless you looked at it last night. But look at this data, and after what I told you about all that you know about DNA, I just told you a lot about DNA that Watson and Crick had figured out. Again, thanks to Rosalind Franklin. When you're looking at this table, what jumps out at you besides the highest and lowest?
It's like a G percentage of both. Humans have 19% or 19.9%.
Okay, so the humans have about the same. Humans have about the same percentages, which makes sense. All right, I'll give you a clue. Compare the A, C, G, and T's. Yeah. So the G has more percentages than what? Oh, the lower percentages. Okay, that's true. Lower percentages, yeah. So what else do you notice? Keep looking. The answer's there in front of you. Yeah. What's all the same numbers? They're all the same numbers? Like, I would have to disagree with that. You're close to the answer, though. Yeah. Speak up. I can't hear you. All I hear is shush, 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 shush.
Oh my gosh. Percentage A does not have percentage.
Percentage A doesn't have, okay, yeah, that's, I'm sorry, ignore that. That is a difference, but you guys are looking for, look at the big picture. Compare the A, the G, the C, and the T. There's something going on. These are all percentages. I'm just getting rid of this because somebody noticed that some of them do have it, some of them don't. I was lazy. Don't even worry about the decimal. What do you notice about these? They're all double digits, okay. That's fair. What do they all have to equal? What do all these have to equal? Say that again? 121? Well, they all have to equal 100%, theoretically, right? So the percentages, they're not going to equal 121 because there's experimental error. Does that make sense? You never get anything perfect. You can't expect things to be perfect. In fact, in science, if things turn out perfect, we question it. We think something went wrong, somebody cheated. Because you shouldn't have any perfect answers. There's always a mistake. There's always some error. So when you're looking at this, and they don't quite add up to 100, but in every organism going across, they should add up to 100. So if you add 31, 32, 18, and 17, and we can round up, 31, 17, 33, and 19, they should add up to 100%. They might be 102, might be 100, it might be 99, but it's close. What do you notice, though, about this data? Yeah. There's a lot of repeat numbers, okay. Comparing A to G to C to T. A and T go down and come back up. Well, that's true. I see that pattern as well. But what about this? What about the fact that A, if you include the error, right? If you include the idea of error, if you just think of it in a big picture, the A pretty much matched the T. And the G pretty much matches the C. They're in the same scale. Like, these are 17, 18, 17 to 20, right? These are 17 to 20. Where these are all in the 30s, 28 to 30. And these are all 28 to 30, right? So if you think about a scale, A matches with T and G with C. That was our first clue on how they base pair. This is where we get the base pairing rules. Chargaff's percentages showed us that anytime you look at any DNA, the percent G always equals a percent C, plus or minus some error. All right? And A always equals a percentage of T, plus or minus some error. The reason is that there's base pairing, right? What we just said up here about DNA. The reason is every time you have an A, you have a what?
C.
Every time you have a C, you have a G. So the percentages need to be equal, or you don't have a double-stranded DNA molecule. Are we all understanding that so far? This was our first clue as to how DNA matched, how base pairs matched. And I think you can answer the rest of the questions. I think they're pretty well based on that. And I hope you watch the movie as they talk about Watson Crick. They're never going to talk badly about Watson Crick because they're famous and people want to think best. And they are. I'm not saying they're horrible people, but they got some real character flaws that you shouldn't idolize anyone, any human, right? And when you're talking about Watson Crick, I hate that they never talk about the negatives, and they really don't talk about Rosalind Franklin enough. I think you'll see here on page 3 of 4, this is her work. That was her picture. That picture took her years to make, and she had to use radiation to make it. She ended up dying. She's a lot like Madame Curie. Have you heard of Madame Curie? Yeah. Madame Curie died of cancer because of all the radiation she was exposed to. And thanks to her and thanks to Rosalind Franklin, we have a lot of really good information that we didn't have without them. But Rosalind Franklin also died of breast cancer. And because she died of breast cancer, she did not get the Nobel Prize. Because you don't get the Nobel Prize if you're dead. And Watson Crick stole this picture from her, and unfortunately she couldn't get credit for it. So remember that when you're looking at this, watching the video. Did that help answer all your questions? All right. Okay, so I have to quickly take attendance. I'm assuming it's time to leave because everybody's wrapping up.
Yeah, we've always submitted. is.
All right, so that's good. We got enough time for attendance. I hate rushing attendance.
That is sucks.
Yeah, but it's a good chance for me to make sure I know your names, which is not always easy.